Okay, good morning everyone. So today is the 3rd of March. It is a Wednesday, and we are looking at enzymes today, so B3.4. So we are still on the biology chapter B3, but we are now moving on to enzymes. So today we are going to define the term catalyst and enzyme, explain how enzymes work as biological catalysts, and justify the lock and key model, relating it to the function of enzymes and explaining how temperature and pH can affect the function of enzymes. So these three objectives will stay for the next lesson as well, which will be factors affecting enzymes. So let's look at the keywords. So we've got enzyme, catalyst, active site, substrate, enzyme substrate complex, lock and key, specific, optimum, collide, denatured, temperature coefficient, metabolism, and complementary. So an enzyme is a biological catalyst which is made up of protein. It is biological because it catalyzes a chemical reaction in living organisms. And on the topic of catalysts, a catalyst is a substance that speeds up a chemical reaction without it being used up in the process. An active site is found in enzymes and catalysts. It is part of an enzyme where a catalyst of a substrate occurs. The substrate is a substance acted upon by an enzyme in a chemical reaction. The substrate molecules are exchanged into product molecules. An enzyme substrate complex is a complex formed from a substrate bound to an active site of an enzyme, for example here. So you've got the substrate and it is complementary because it is the same shape as the active site. Lock and key hypothesis. This is a mechanism proposed to explain the binding between active sites of an enzyme and the substrate. Just like only one key can open a lock, only one type of enzyme can speed up a specific reaction. So enzymes are very specific to the substrate that they catalyze the reaction to. Specific, following on from that, enzymes are specific, they act on only one substrate. Optimum, conditions that produce the best results possible. Collide means that they bump or knock into each other. Denatured is the state of a protein when its shape has been altered permanently and it can no longer carry out its function. An important thing to note is do not say that enzymes die, they are not living organisms themselves. Temperature coefficient, or Q10, this is a measure of how the rate of reaction changes or a biological or chemical system as a consequence of increasing the temperature by 10 degrees Celsius. A Q10 of 2 tells you that the rate doubles when temperature is increased by 10 degrees Celsius. If it is free, if Q10 is free, it trebles. And metabolism is the sum total of chemical processes that occur in living organisms. So what is a catalyst to start off? So a substance that speeds up the rate of reaction of the rate of a chemical reaction but remains unchanged in the process of the reaction. So hydrogen peroxide is broken down into water and oxygen by manganese oxide. So that is the catalyst for this reaction. That is what breaks that down into its products. So hydrogen peroxide is the substrate, manganese oxide is the catalyst, and water and oxygen are the products of this reaction. So there we go. We've got hydrogen peroxide, and it's broken down into smaller molecules with water and oxygen. So biological catalysts then. Enzymes act as biological catalysts. They occur inside cells or are secreted by cells. So you would remember from the previous lesson where we talked a little bit about enzymes and things like that, about saliva and amylase. That is what those are. So they are biological catalysts, and they are, if they're in the salivary glands, they're secreted by the salivary glands, and they work on starch to break it down into smaller molecules. So here we have an example. So we've got catalase, and it breaks it down into smaller molecules. So enzymes are also proteins. So enzymes are made of a protein. You would have seen this in the lesson on proteins. And as you know, protein synthesis occurs in ribosomes. Proteins are long molecules of amino acids that are folded into a very specific 3D shape. And all enzymes have A's on the end. So ASE. Anything that has A's on the end, it will be an enzyme. For example, protease, amylase, lipase, and carbohydrates. They all have A's on the end. That was the dog. So they all have A's on the end because they are enzymes. So that is how you can see that they are enzymes if you look at the name of them. So enzymes, the part of the enzyme where the reaction occurs is known as the active site. The substrate molecules go into the active site and they are complementary and fit the active site perfectly, which is your lock and key mechanism. And the active site is the site on the enzyme where the reaction occurs, and this big purple blob is the enzyme itself. So these white bits are the active site, and the molecules are the substrate molecules. So these are your product molecules that are made by the enzyme catalyzing the reaction. So enzymes are reusable. They can be used over and over again, like catalysts can. So the lock and key hypothesis states that the active site must be complementary to the shape of the substrate molecule, and the rate of an enzyme-controlled reaction is affected very much by temperature, especially by an increase in temperature. What do enzymes do then? So enzymes make larger molecules from lots of smaller ones, including making starch, glycogen or cellulose from glucose, lipids from fatty acids, and proteins from amino acids. They can change one molecule into another, for example, changing one simple sugar into another, for example, glucose into fructose, or one amino acid into a different amino acid. They can also, on the flip side of this one, they can break down larger molecules into smaller ones. So breaking down carbohydrates, lipids, and proteins into their monomers during digestion, glucose in respiration, and excess amino acids to form urea, which is your waste product. So just a quick test of your understanding then. This is a very short lesson in comparison to the other ones, but I'd just like you to identify which of these you think are true or false. So write down these statements and then write next to them whether they are true or false. So enzymes increase the energy needed for a reaction to take place, true or false. Enzymes are chemical catalysts, true or false. Enzymes remain unchanged after a reaction. Enzymes are protein molecules. Enzymes can build larger molecules from smaller ones, and enzymes and their substrates have the same shape. So after you've done that, we have some extension questions here, which are tiered using the bronze, silver, gold. So the first one is define the term macromolecule. Second one is to give 3 clear examples of the type of reactions that are catalyzed by enzymes. So they will be reactions that happen in the body. And the third one is to explain in detail how enzymes act to speed up reactions in your body. And the final thing I'd like you to do is watch this video on YouTube, and that is the manganese experiment, and it will show you how to do that because we can't do it in the lab because we are not in school. Watch the video, and then I have attached a worksheet to Microsoft Teams, the assignment on Microsoft Teams that I'd like you to complete.